The flukes clear the surface just after the exhale. A blow angled forward and to the left, the angle distinctive to a sperm whale and no other species. And then 50 tons of animal turns its head down. The ocean closes over it. That breath is now the only oxygen available for the next 45 minutes. Below 200 meters, sunlight fails. Below 1,000 meters, NOAA classifies the environment as the midnight zone, a name that owes more to accuracy than poetry. Sperm whales have been directly tracked by telemetry, reaching foraging depths of around 2,000 meters, with the deepest instrumented individual documented at approximately 2,250 meters. The absolute mammalian record belongs to a different species. Schorr and colleagues documented a Cuvier's beaked whale reaching 2,992 meters in 2014. At the depths where sperm whales routinely hunt, pressure already exceeds 200 atmospheres. At 3,000 meters, a depth representing the outer envelope of claimed capability for the species, it reaches approximately 300 atmospheres, equivalent to roughly 4,400 pounds pressing inward on every square inch of surface. An air-breathing mammal hunts here on one breath for roughly 45 minutes per dive. That figure isn't a record. Multiple independent datasets converge on it as the average. Watwood and colleagues, in a 2006 Journal of Animal Ecology paper drawing on tag data from multiple ocean populations, recorded average dive cycles of 45 minutes submerged and 9 minutes at the surface. A Mediterranean study by Drouot and colleagues found 44.8 minutes on average. Maximum documented durations from acoustic tracking data going back to Watkins and colleagues in the 1980s exceed 130 minutes. What we're looking at isn't exceptional performance. It's a working day. The question isn't how a sperm whale tolerates extreme depth. The question is what exactly transforms inside this animal the instant it submerges, and why every part of it transforms simultaneously. The first transformation begins before the whale has passed 100 meters. The mammalian dive response is universal, a reflex present in every air-breathing vertebrate, triggered by immersion and the cessation of breathing. But in large diving cetaceans, the scale of it sits in a different register entirely. In 2019, Jeremy Goldbogen and colleagues at Stanford achieved the first direct ECG measurement of heart rate in a free-swimming large whale, attaching a biologging tag to a blue whale in the Pacific. Surface resting heart rate averaged between 25 and 37 beats per minute. At the bottom of a foraging dive, it fell to between 2 and 8 beats per minute, with individual readings as low as a single contraction every 30 seconds. Sperm whale heart rate hasn't been directly measured by ECG tag in free-diving animals. The technical challenge hasn't yet been solved, but the mechanism is identical across cetacean species. Vagal nervous system activation in response to apnea, producing bradycardia proportional to the anticipated dive. Research on harbor porpoises, published in 2016, found that these animals modulate their heart rate before they submerge, calibrated to how long they expect to be down. The body begins managing the budget before the first fluke stroke. Slowing the heart reduces the oxygen consumed by the cardiac muscle itself. It also reduces cardiac output, which limits blood delivery to peripheral tissues, and that is what triggers the body's deeper triage. Vessels supplying the skin, gut, and locomotory muscles narrow or close entirely. The digestive system goes offline. The tail muscles, driving the whale deeper with each stroke, are cut off from the central circulation. They will receive no fresh oxygenated blood until the animal surfaces. For the full duration of the dive, those muscles run on internal reserves alone. The architecture for those reserves is where the actual story lives. Koyman and Pongamis established in their 1998 Annual Review of Physiology analysis that a 10,000 kilogram sperm whale carries approximately 77 milliliters of usable oxygen per kilogram of body weight. For context, a human being manages around 27 milliliters per kilogram. The whale carries nearly 3 times as much oxygen, mass for mass. The distribution matters as much as the total. In humans, the lungs hold the majority of onboard oxygen. In a sperm whale, the lungs account for only around 10%. Blood holds approximately 58%. Muscle tissue holds the remaining 34%. Most of the oxygen is already positioned in the tissues that will need it most, so that when the central circulatory supply is closed, those tissues don't immediately starve. That muscle oxygen is stored in myoglobin, a small iron-containing protein that binds one oxygen molecule per unit and releases it as local oxygen pressure drops. In human skeletal muscle, myoglobin concentration averages 4 to 7 milligrams per gram of wet tissue. Across the toothed whale family, direct measurements by Ponganis and colleagues in the Journal of Experimental Biology in 2000, across 10 odontocete species, found concentrations from 18 to nearly 58 milligrams per gram. Deep water species cluster toward the top of that range. Sperm whales, as the largest toothed whale and the species most committed to depth hunting, are expected at the ceiling. The result is visible to anyone who has seen the flesh. Sperm whale swimming muscle is very nearly black, not red, black. That color is the myoglobin, so densely packed into the cells that it dominates the tissue's appearance entirely. Stability is what permits density. Human myoglobin aggregates into dysfunctional clumps at concentrations that whale myoglobin tolerates comfortably. The cell can only produce as much myoglobin as it can keep properly folded. Masita and colleagues, in a 2013 paper in Science, traced the molecular mechanism of this across the cetacean lineage. Diving mammals, independently across seals, sea otters, and cetaceans, accumulated positive surface charges on their myoglobin molecules, causing individual protein units to repel each other electrostatically. This prevents aggregation. Ancestral cetaceans, reconstructed from sequence data, had myoglobin similar to their terrestrial relatives. Sperm whale myoglobin has substantially more positive surface charge. The same adaptive pressure, arriving through different molecular pathways in different lineages, converged on the same solution. Charge the protein surface so more of it can be packed into each cell without clumping. The protein has been under intense selective pressure precisely because it's so central to breath-hold capacity. So the muscles run on their black myoglobin stores, isolated from the heart's circulation, working aerobically as long as the local oxygen supply lasts. When it's spent, the cells switch to anaerobic metabolism. Lactic acid accumulates, but in the isolated tissue, contained by the closed vasculature, it has nowhere to go. It stays in the muscle until the whale surfaces and blood flow is restored. Then it floods into the circulation. A post-dive spike in blood lactate, a temporary drop in blood pH, the measurable signature of a dive that pushed past the aerobic limit. The debt is real, it's simply paid later. That oxygen problem has a companion: pressure. At 1,000 meters, hydrostatic pressure is approximately 100 atmospheres. At 2,000 meters, 200. At 3,000 meters, 300. Pressing inward simultaneously from every surface of the animal's body. Any structure that depends on a sealed gas volume to maintain its shape is threatened. The sperm whale's lungs aren't designed to resist. They are designed to yield. The rib cage attaches to the spine and to a multi-segment sternum through flexible cartilage, not fused bone. The sternum itself is composed of multiple bony sections joined by cartilage, not a single rigid structure. This allows the thoracic cavity to compress inward as external pressure increases, rather than resist it. The collapse of the alveoli, the small gas exchange surfaces where oxygen passes into blood, is progressive, advancing with depth, beginning at relatively shallow depths and completing at deeper ones. A 2023 review of cetacean respiratory anatomy, published in the Anatomical Record by Reidenberg and Laitman, confirmed that this thoracic architecture is a defining adaptation across diving whales. The chest is explicitly protected from pressure damage precisely by being able to give. Once the alveoli collapse, gas exchange across the lung surface stops. No more nitrogen enters the bloodstream. The mechanism that causes decompression sickness in human divers, nitrogen dissolving into blood under high partial pressure, then forming bubbles on rapid ascent, simply can't operate if the alveoli are already flat. A 2012 analysis in Frontiers in Physiology, examining decompression risk in diving cetaceans directly, identified alveolar collapse as the primary protection against nitrogen loading during deep dives. The compressed air is displaced into the rigid conducting airways, the trachea and bronchi, where no exchange occurs. It's stored, not lost. The sinuses and air spaces within the whale's head participate in pressure management as well. These cavities are lined with dense networks of blood vessels that engorge with blood during descent, physically flooding the air spaces. Where there was a gas-filled cavity, there is now blood. Structures that could implode under 300 atmospheres become blood-filled chambers. The body systematically eliminates every gas volume that would otherwise become a pressure failure point. The animal that enters extreme depth is one that yields rather than resists at every level. Below 1,000 meters, with lungs collapsed, no circulation reaching the muscles, and total darkness, the whale still has work to do. That work requires one system still running at full capacity. The sperm whale's head, comprising roughly a third of total body length in large adult males, is built around a problem: how to locate moving prey in complete darkness. The mechanism that emerged, refined across 50 million years, is unlike anything in any other animal. Air is pressurized and forced through a pair of specialized membranes in the right nasal passage, structures called the phonic lips, or museau de singe in anatomical French, monkey's lips. As air passes between them, they collide and produce a sharp click. That pulse travels backward through the spermaceti case, a large oil-filled structure running the length of the upper head, strikes a gas-filled frontal sac, reflects forward, and passes through a series of oil-filled lenses called the junk. The junk concentrates the pulse into a narrow forward beam. The result was directly measured by Merle and colleagues in a 2003 paper in the Journal of the Acoustical Society of America. Source levels up to 236 decibels, reference to 1 micropascal at 1 meter, measured in water. Their description of this measurement: by far the loudest of sounds recorded from any biological source. Comparisons to aircraft engines that circulate in popular accounts are acoustically misleading. Underwater measurement uses a different reference pressure than airborne measurement, making direct numerical comparison meaningless. But the biological record stands on its own. No other animal generates instantaneous sound pressures at this level. The click beam travels outward through water and strikes prey. A squid, most likely, or a deep-water fish. The echo returns. From the round-trip travel time, the whale calculates distance. A 2020 tagging study by Tunison and colleagues, using hydrophones mounted directly on a sperm whale's rostrum to capture echoes as the animal heard them, recorded individual prey detections at ranges up to 144 meters and school-level detections at around 245 meters. In absolute darkness, using nothing but reflected sound, the whale is tracking moving targets the length of 2 football pitches away. As the prey closes within striking range, the click rate accelerates. Inter-click intervals shorten from around 500 milliseconds to less than 100, producing the dense, rapid-fire burst researchers call a creak or buzz. Madsen and colleagues, in a 2016 Scientific Reports paper examining predator-prey interactions directly, found that source level during the buzz phase drops by 1 to 2 orders of magnitude compared to normal foraging clicks. This rules out the popular account that sperm whales stun prey acoustically. The buzz isn't a weapon. The reduced power is incompatible with acoustic incapacitation at that range. It's precision targeting at close quarters. The whale refining the geometry of capture, not delivering a killing blow with sound. The ascent begins. Lactic acid trapped in the isolated muscles enters the general circulation as vasoconstriction eases. Blood pH drops transiently. The heart accelerates back towards surface rates. The lungs re-expand as pressure falls with each meter of ascent, and gas exchange across the alveoli gradually resumes. 9 minutes at the surface. The Watwood 2006 dataset is consistent across multiple ocean regions. 9 minutes average surface interval, 3 minutes standard deviation. In those 9 minutes, oxygen loads back into hemoglobin and myoglobin. Lactic acid is metabolized. Cardiac function returns to resting parameters. More than 72% of the sperm whale's time is spent in the dive phase. Those 9 minutes aren't recovery. They are the reload before the next run. Then the flukes rise again. The sperm whale survives at extreme depth, not because any single adaptation is exceptional enough in isolation, but because 5 systems shift state together, timed together, managed in concert, before the first fluke stroke has pulled the animal a body length below the surface. The heart slows. Blood is rerouted away from periphery and toward brain and cardiac muscle. The myoglobin-loaded, near-black swimming muscles run on internal oxygen stores, isolated from the central supply, carrying the aerobic load as long as possible and the anaerobic load beyond that. The chest compresses progressively inward, eliminating nitrogen uptake and pressure-vulnerable air volumes simultaneously. And in total darkness, a click beam fires forward from the most complex acoustic structure in the animal kingdom, finding prey by the return time of sound. Fail any one of those mechanisms, and the dive fails. Keep all of them operating in concert, and a 50-ton air-breathing mammal reaches the deepest hunting grounds of any predator in the mammalian lineage, and surfaces 9 minutes later, ready to go again. Which part of that system should we look at next? The heartbeat, the lungs, the oxygen storage, or the clicks? Leave your answer in the comments. Production disclosure. This script was developed with AI assistance in research synthesis and drafting. As an observational note from a sample of 50 visible comments audited on this channel, 14 of those comments referenced perceived AI involvement, generating 44 instances of engagement on that point. That figure reflects the 50 comment sample only and isn't statistically representative of the full audience.